Now we can begin talking about the four major classes of macromolecules found in biological systems. And they are the carbohydrates, the nucleic acids, the proteins, and the lipids, or fats. No, let's call them lipids actually, because fats are a subclass of lipids. So we have carbohydrates, nucleic acids, proteins, and lipids. We can talk about how these polymers are built up, what subunits are involved in, that is what monomers are involved in being linked together to form these various macromolecules that are the carbohydrates, nucleic acid, proteins, or lipids. So for carbohydrates, if we look at the types of carbohydrates that are common, there are others, but let's just look at these. We have starch and glycogen, which are the storage forms of carbohydrate in animals and plants. Plants for starch, glycogen in animals. And the subunit is a sugar molecule called glucose that we'll look at. Glucose is polymerized into starch or glycogen. And then when glucose is needed, the starch or glycogen is broken down into glucose, which can then be fed into the metabolic pathways to generate the energy currency of the cell, ATP. We'll talk about that later. So glucose, starch and glycogen for energy storage. An example, potatoes. Potatoes are full of starch. Beets are another example. The roots of major plants are storage organs basically for starch, which can be used for energy later. Cellulose is also a polymer of glucose, glucose being the monomer. And cellulose is one of the most abundant macromolecules on Earth, if not the most. And it forms the cell walls, a major component of plant cell walls. And paper, strings of celery, the cellulose that you find in celery, the reason it's so fibrous is because of cellulose. We'll talk more about that as well. Now, chitin is a modified glucose molecule that is polymerized to form chitin, a type of carbohydrate that is used for structural support for the exoskeletons of invertebrates, of insects and crabs and lobsters and things like that. So chitin is an important structural support. Chitin is also found in fungi, mushrooms, interestingly enough. So both invertebrates and fungi have seized upon chitin as the skeletal support for them. It's a polymer of modified glucose. Now the nucleic acids, named because they are acidic due to phosphate groups that we'll talk about, and they're found in the nucleus of cells. So the nucleic acids, DNA and RNA. And the building blocks for DNA and RNA are nucleotides that are polymerized to form RNA or DNA, depending on the nucleotides that are being used. DNA is the stuff of genes, and those genes carry the instructions to build organisms and run the biochemistry of the cell. And RNA, as we will see and study, is needed for gene expression. Examples, DNA is found in chromosomes, and RNA, a type of RNA called messenger RNA, is used as the messenger that links the gene to proteins. And we'll talk about the coding and decoding of genetic information. We will see that messenger RNA is very important. Now, proteins. We can have functional proteins or structural proteins. They're both polymers of amino acids that are linked together. For functional proteins, there are many things that proteins can be involved in. They can be enzymes that promote biochemical catalysis and run the biochemical pathways in the cell. They can be used, proteins can be used for transport. Proteins can be used as defense mechanisms. The immunoglobulin proteins that are an important part of our immune systems, our response to the invasion of our bodies by foreign substances. Those immunoglobulins are proteins. There are proteins that are regulatory proteins that bind to DNA and turn genes on or off. There are storage proteins. Proteins can be used to store energy, although usually lipids or carbohydrates are used for that function. And proteins can also be used in transport, as we will see. Proteins form molecular motors that move substances within cells. And proteins also can form rigid support for in structures of the cell, the cytoskeleton that we'll talk about. So examples, for example, of structural proteins: hair, silk, the actin microfilaments that we'll find in cells that are part of the cytoskeleton. An example of a functional protein would be hemoglobin, which carries oxygen around in our bloodstreams. Now lipids, the fourth major class of macromolecules. We have fats, phospholipids, prostaglandins, steroids, terpenes as examples of the lipids there. Fats are used for energy storage, and the subunits, the monomers that are linked together to form fats, are glycerol and 3 fatty acids that are linked together with the glycerol. We'll talk more about that shortly. Examples: butter, corn oil, soap. Phospholipids are used in membranes. They are the major component of biological membranes, the plasma membrane of cells, as well as the subcellular compartments in eukaryotic cells that we'll mention. Now, phospholipids are made of glycerol, as are fats, but linked to that glycerol are 2 fatty acids, not 3, and a phosphate and another polar group of atoms linked together, a polar functional group. Those constitute phospholipids, and we'll talk a lot about those. We're going to spend a lot of time talking about membranes and the importance of membranes for cellular life. Now, prostaglandins are chemical messengers, like prostaglandin E, for example. And prostaglandins are 5 carbon rings with nonpolar hydrophobic tails on them, very important signaling molecules. Steroids also are used as signaling molecules. They are hormones like estrogen and testosterone, the primary sex hormones. But steroids can also be components of membranes. They can fit into the phospholipid bilayers that constitute membranes, as we will see. And steroids then there are 4 fused carbon rings joined together. Those identify the steroids. And finally, terpenes are a type of lipid that are very long carbon chains, and they function as pigments or structural support, especially in plants. And examples are keratin, the keratin that causes carrots to be orange. Keratin is a pigment, and keratin is also a precursor to some vitamins used in animals that animals need. And rubber, natural rubber. Now we all have synthetic rubber, but in the old days, rubber was all natural, derived from rubber trees. And rubber is basically an example of polymer, long carbon chains polymerized into terpenes. So now we're going to consider 2 types of key biochemical reactions that are used to polymerize monomers into polymers, and then the reaction that depolymerizes polymers into individual building blocks or monomers. So first, dehydration synthesis is used to polymerize many the polymers that we find. So for example, here are 2 molecules that are being linked together, and this carbon is being joined to that carbon, and what's being removed is 1 hydrogen, 2 hydrogens, and an oxygen, and which form molecular water. So a dehydration reaction, that is, we're removing water, we're dehydrating. It's also known as condensation reaction, just like if you have your breath, water condense, you breathe out onto a cold surface, your breath will condense onto that surface. That's water condensing on the surface. So dehydration reactions or condensation reactions remove water and are used often to polymerize subunits into polymers. Whereas hydrolysis, that is the addition of water, hydro meaning water, lysis meaning to break up, water can be added to break up a polymer into individual building blocks, into individual monomers. So hydrolysis is the opposite of condensation or dehydration reactions. And These are very important in the polymerization of macromolecules and the depolymerization of macromolecules. Water is heavily involved in that. So you'll want to get that straight. Dehydration is used in synthesis. Hydrolysis is used in breaking down. So now let's move on to talking specifically about carbohydrates. We'll now treat all the 4 classes individually: carbohydrates, nucleic acids, proteins, and lipids. And we'll speak about those in each in a bit of detail. So let's start with carbohydrates. So carbohydrates are characterized by having a particular ratio of carbon, hydrogen, oxygen. That's what constitutes a carbohydrate. Namely, 1 carbon to 2 hydrogens to 1 oxygen. So you could just say this is CH2O times how many ever repeats of this particular empirical formula there is. So 1 to 2 to 1 ratio carbon, hydrogen, oxygen. And as you can see then, there's a lot of carbon and hydrogen present, a lot of CH bonds, and those carbon-hydrogen bonds are high energy bonds. They can be broken down to derive energy from them. And that's why carbohydrates are good storage molecules for energy. So as I've said, the CH bonds hold a lot of energy, and therefore carbohydrates are good energy storage molecules. Like sugars. Sugars are carbohydrates. Starch is a storage form of sugars linked together. Glucose is a single sugar. So let's talk. We'll come across these again. Let's talk about some of these carbohydrates. Let's look at some monosaccharides. Mono meaning 1, saccharides meaning sugars. So let's look at single-ringed sugar. 6 carbon sugars, like glucose. There's 6 carbons, twice that number of hydrogens, and 6 oxygens, 1 to 2 to 1 ratio. And fructose, fruit sugar, the sugar found primarily in fruit, is an isomer of glucose. It's a structural isomer of glucose, as we will see. Whereas galactose is a stereoisomer. We've already talked about that, actually. We'll revisit it briefly. Now, the enzymes that are present, the protein enzymes that break down sugars or that link sugars together into starches, can recognize the different isomers of the 6 carbon skeleton. So this is where isomers are very important. Some of them are recognized by one enzyme, other isomers are recognized by other enzymes. Now starting the next section, we'll talk about these different isomers of a molecule glucose. We'll talk about isomers of monosaccharide glucose and look at the difference between those isomers. And then that will set up later talking about how those different isomers of glucose are utilized to form different starches.